Now let's zoom in on bioavailability, which is an important parameter for every dosage form. Bioavailability is the rate and extent to which a drug is absorbed and becomes available at the site of action. For drugs administered by the intravenous route, bioavailability is 100% because the drug is injected directly into the vein and therefore all of it enters the blood directly. There are many factors affecting bioavailability. Physiological factors are related to the human body. The surface area of the absorptive site affects bioavailability. The larger the surface area, the larger the amount of drug that can be absorbed. For example, the gastrointestinal tract provides a very large surface area for the absorption of drugs. The blood flow to the site of administration also affects bioavailability. If the area has many blood vessels, the drug will be absorbed into the blood more quickly. For oral administration, a number of factors affect bioavailability, such as the pH of gastrointestinal fluids. pH affects the absorption of weakly acidic and basic drugs. If you still remember, the unionized form of the drug will be better absorbed because they can cross lipid membranes more easily. Gastric emptying rate is the rate at which the stomach empties its contents into the small intestines. Most drugs are absorbed at the small intestines, so any delay in gastric emptying time will slow the rate of drug absorption. Intestinal motility is the movement of the intestine. For optimum absorption, the drug must have sufficient time at the absorption site. If there is very high intestinal motility, for example in diarrhea, the drug has a very brief time in the intestines, which results in lower opportunities for absorption. The first pass effect is the metabolism of the drug by the liver before it reaches the systemic circulation. The presence of food in the gastrointestinal tract can also affect bioavailability. The effects of food are not always predictable. Some drugs should be taken with food, others should be taken on an empty stomach. Physical-chemical properties of the drug itself can also affect bioavailability. The drug dissociation constant, Ka, and lipid solubility affects how well a drug can cross biological membranes, which is crucial for absorption. Dissolution rate: the faster the dissolution rate, the faster the drug can be absorbed. Chemical stability is also important. If the drug is destroyed by stomach acid or enzymes, then it would have a poor oral bioavailability. Some drugs may form complexes with or be adsorbed onto other contents in the gastrointestinal tract. For example, some antibiotics can form complexes with iron and calcium, which hinders their absorption. So you would find instructions on the label to avoid taking calcium and iron containing products together with the medication. The dosage form also affects bioavailability. Excipients can affect how the drug is released and therefore affect bioavailability. Different types of dosage forms also have different bioavailabilities. In general, aqueous solutions will be absorbed the fastest, followed by aqueous suspensions, hard gelatin capsules, uncoated tablets, and then coated tablets. Here we see an example of how different dosage forms of a drug have different bioavailabilities. In this example, 800 mg of albendazole is administered in different dosage forms. The cyclodextrin solution has the highest bioavailability as it provides the highest plasma drug concentration. The suspension has a lower bioavailability, followed by the tablet. The suppository was not able to deliver the drug at all to the systemic circulation. To conduct a bioavailability study, the drug is administered to healthy volunteers and their blood is regularly sampled for concentration testing. Plasma drug concentration time curves are constructed using the data. The absolute bioavailability of a drug is the fraction of the administered dose which is absorbed intact into the systemic circulation. The bioavailability of the IV dose of any drug is 100%. Therefore, the IV drug can be used as a reference to calculate the bioavailability of a dosage form administered by any other non-IV route. So the dosage form will be administered to healthy volunteers, and the plasma drug concentration time curve will be constructed. So you can see this here, the plasma drug concentration time curve for the dosage form. The same thing will be done using the IV route to administer the drug. And you can see this is the curve for the IV route. The area under the curve represents the amount of drug absorbed into the blood. Therefore, absolute bioavailability is calculated by taking AUC of the studied dosage form divided by its dose over the AUC of the IV drug divided by the IV dose. Sometimes we want to compare the bioavailability of a dosage form to another non-IV dosage form. This is called relative bioavailability. A comparison is usually made to a standard dosage form which is already known to be well absorbed. The plasma drug concentration time curves are constructed for the test dosage form as well as the standard dosage form. The relative bioavailability is calculated by taking the AUC of the test dosage form divided by its dose over the AUC of the standard dosage form divided by its dose. Let's do a practice calculation. Let's say a study was undertaken to study the bioavailability of a new drug formulated as a tablet and a capsule. The AUC calculated after the tablet A of 150 mg was given was 50 mg hours per liter. After an oral capsule dose B of 75 mg, the calculated AUC was 25.3 mg hours per liter. Calculate the relative bioavailability of the tablet A with respect to the capsule B. Press pause to do your own calculation first, then press play to review the answer. The relative bioavailability is AUC of the test dosage form divided by its dose over the AUC of the standard divided by its dose. Here it says calculate the relative bioavailability of tablet A with respect to the capsule B. So tablet A is the test dosage form, while capsule B is the standard. Plug in the values. AUC of tablet A is 50, the dose is 150. AUC of tablet B is 25.3, the dose is 75 mg. Therefore, the relative bioavailability of tablet A with respect to capsule B is 0.988. There are a number of examples of drug products which have exhibited bioavailability problems in the past. These examples are all pre-1976. You don't really hear of such problems nowadays, and this is an indication that more attention is now being given to bioavailability studies. Chlorpropamide is an anti-diabetic drug. Here we see that when 3 products were tested, the peak plasma concentration for brand C was only half of the peak of the 2 other products A and B. Digoxin is a medication used in heart failure. Doctors in Israel noticed 15 cases of digoxin toxicity between October to December 1975. This was unusual since there were almost no reports for the same period the previous year. It was later found that the local manufacturer had changed the formulation to improve dissolution without telling the physicians, and this change resulted in a 2-fold increase in the availability of the new formulation. So essentially, patients were getting 2 times the dose. Phenytoin is an anti-seizure medication which can also result in many side effects. One report described an incidence of phenytoin intoxication in Australia in 1968 and 1969. Apparently, the tablet diluent was changed from calcium sulfate to lactose. Later studies showed that the bioavailability was higher from the dosage form containing lactose. Let's look at some terms that are used for similar drug products. Drug products that contain the same amounts of the same active ingredients in the same dosage forms that meet the same or comparable standards of strength, quality, purity, and identity are termed as pharmaceutically equivalent dosage forms. However, pharmaceutical equivalence does not necessarily imply bioequivalence. Differences in excipients and manufacturing process may lead to differences in dissolution and absorption between the products. So, what is bioequivalence? The two dosage forms must have the same dose of the same drug, and there should be no significant difference in their peak concentrations, times of peak concentrations, and areas under the curves. Before a generic product can be approved to be marketed, the manufacturer must first submit data to demonstrate that the generic drug product is bioequivalent to the innovator drug product. Therapeutically equivalent drugs are those that generally are considered most appropriate for substitution. According to the FDA, a therapeutically equivalent generic product can be substituted for the branded product. To be therapeutically equivalent, an important criteria is that the products must be bioequivalent. So, bioequivalence studies are performed to compare the bioavailability of drug products. If two medicines are bioequivalent, it means there is no clinically significant difference in their bioavailability. Bioequivalent products would have very similar peak concentration, time to peak concentration, and area under the curve. Most of the time, bioequivalence studies are performed for generic drugs to compare it with the innovator product. Bioequivalence is taken as a surrogate marker for clinical effectiveness and safety data. The innovator company has already done the clinical trials to prove the safety and efficacy of the drug, so it wouldn't be practical for the manufacturer of generic products to repeat the clinical trials. If they can demonstrate bioequivalence, it is assumed that the generic product will have similar efficacy and safety as the innovator product. Bioequivalence studies may also be conducted by innovator companies to compare formulations. For example, to compare formulations used in the clinical trials with the formulation for the product to be marketed. Bioequivalence studies may also be conducted when there is any change in formulation, for example, a change in one or more excipients. In this section, we've looked at the definition of bioavailability, factors affecting bioavailability, and how to calculate absolute and relative bioavailability. We've also looked at the difference between pharmaceutical equivalence, bioequivalence, and therapeutic equivalence. For bioequivalence studies, the Cmax, Tmax, and AUC of the two products needs to be similar.